and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about northern blot. The northern blot is a technique that is useful to study gene expression by detection of RNA or isolated mRNA in a sample. The process of northern blotting involves the separation of RNA by gel electrophoresis and detection of RNA with a hybridization probe. The northern blot is also known as RNA blot. Strictly speaking, the term northern blot refers specifically to the capillary transfer of RNA from gel to a blotting membrane. However, commonly the entire procedure is known as northern blotting. The technique of northern blotting was developed by three scientists, namely James Irwin, David Kemp, and George Stark at Stanford University in 1977. This technique is known as northern blotting because of its similarity with another blotting technique known as southern blotting. Next, we shall discuss about various steps involved in the process of northern blot. In northern blot, first step is the extraction of total RNA from a homogenized tissue sample or cells. After extraction of total RNA, the eukaryotic mRNA is isolated by the process of oligo-DT cellulose chromatography. In this chromatography, only the RNA that has poly tail will bind to the oligo-DT cellulose column, by which they can be isolated. After the isolation of eukaryotic mRNA, the RNAs are separated by the process of gel electrophoresis on the basis of size. You see here, this is a gel in which these are the RNAs which are separated on the basis of size. After separation of RNA, the RNA are transferred to a nylon membrane by the process of capillary blotting or vacuum blotting system. The nylon membrane, which is positively charged, is most effective for this process because the negatively charged RNA molecules will have strong affinity for this nylon membrane. And the transfer buffer that is used during the process of blotting contain formamide. Formamide reduces the annealing temperature of RNA probe interaction, thereby preventing the use of high temperature which can degrade RNA. After the transfer of RNA from gel to the nylon membrane, next step is the immobilization of RNA. Immobilization of RNA is carried out through covalent bonding by UV light or heat. And after immobilization of RNA, the next step is to hybridize the RNA with a suitable label probe. After hybridization with a label probe, the nylon membrane is washed to ensure that the probe has bound specifically and there is no background noise. After washing, the signal can be detected by X-ray film and the RNA can be quantified by the process of densitometry. This is the process of northern blot. Next we will see that what type of gel is useful in northern blot. The agarose gel electrophoresis is commonly used for the process of northern blotting. The agarose gel, which contain HCHO, that means formaldehyde. The formaldehyde, it limit the formation of secondary structure of RNA. And the agarose gel can also be stained with ethidium bromide. By staining with ethidium bromide, we can determine the quality and quantity of RNA. Similarly, one can also use polyacrylamide gel electrophoresis to separate RNA during the process of northern blot. But polyacrylamide gel electrophoresis is most suitable for segmented RNA or microRNA. Next is probes. Probes are nucleic acid with a sequence that is complementary to the entire target RNA or a part of the RNA. The probe can be RNA, DNA or a short oligonucleotide that contain 25 complementary nitrogenous bases. There is another type of probes known as riboprobes. Riboprobes are RNAs which are transcribed in vitro. This type of probes are called as riboprobes. Probes can be labeled with radioactive isotope, for example, phosphorus 32. And the probes can also be labeled with chemiluminescence, in which an enzyme, for example, alkaline phosphatase or horseradish peroxidase, break down a chemiluminescent substrate, thereby emitting light. And this signal can be detected by X-ray film. So this is the process of northern blot. Then we will discuss about the application of northern blotting. The RNA blot is useful to study the expression of particular gene between tissues, organs, developmental stages, environmental stress level, and during pathogen infection, and also over the course of treatment. With northern blot, we can also show the overexpression of oncogenes and downregulation of tumor suppressor gene in cancerous cells. This is about northern blot. Please share, like and subscribe this video, and thank you. Bye.